All right, so this unit is on DNA, the history, structure, and replication. So, how do we make new DNA? What I've tried to do with these notes for this unit is mark in green kind of the really important terms or parts of each slide so that you know where to focus on. And so far, this unit we've covered the structure of DNA in class. We've built the DNA molecule, and we have gone over kind of the history to the discovery of this molecule. So here's some of the important people. We're going to cover most of the ones that we covered on the stations lab, but we're going to leave out Hershey and Chase, who did the little virus thing. They basically confirmed what Avery discovered with DNA being the stuff that our genes are made of. And so Frederick Griffith, if you remember from the stations lab, worked with mice. And basically, he injected mice with pneumonia, uh, some that would cause death and some that would not cause death. But the key thing that he discovered is that when he mixed the two together, something was being transferred. So he called it a factor. But basically, understand that DNA, or this factor, this transforming principle, was moved from one bacterial cell to another. Um, we then move on to Oswald Avery, and he's really the guy that said DNA is the stuff. So he looked at Frederick Griffith's paper and work and said, okay, if I start to take out the different pieces of the cell, if I eliminate all the protein, if I eliminate all the lipids, which is where the lipase is down here, if I take out all of the RNA, what happens to this mouse? And every time he ran this experiment, the mouse died. The mouse died. The mouse died. The only time the mouse didn't die is when he removed DNA. And so basically he concluded that DNA was the stuff, was this factor or transforming principle. That was later confirmed with Hershey and Chase and what they did with those radioactive viruses. Moving on to like the 1950s, you have Chargaff, who basically discovered that A pairs with T and C pairs with G. And how he did that is he was able to analyze different species, the amounts of adenine, thymine, cytosine, and guanine, and found that if you look at this chart over here for human DNA composition, we're about 32, 33% adenine and about 31, 32% thymine. So he concluded that those two must bond together. Same thing for cytosine and guanine, which looks a little bit more than 20% on this graph. Of the bases, they really fall into two classes. You have the purines, which are double ring, and you have the pyrimidines, which are single ring. The way I remember that is I always think of a pyramid as being one triangle or one square, so I think of that as one ring. And to how I remember the bases, thymine has a Y in it, and so does cytosine, and so does pyrimidine. And so he used that is what he came up with. And kind of here's his rule. He understood how the rungs of the ladder kind of fit together. Next we get to Rosalind Franklin, who basically kind of gets left out from a lot of the discoveries up until recently. But in the 50s, she started to work with X-rays, and it's called X-ray crystallography. And she was able to take a picture of a single strand of DNA. You can see it here. These black marks are where the DNA is touching the X-ray screen, and so this showed that it was spiral, or like think of like a slinky. If I stretch the slinky out, the length of this, only where the slinky is touching makes a black mark. This was really a breakthrough discovery. Um, she didn't know exactly what she had in terms of how to build the molecule. She knew it was somewhat of a spiral. And so that leads us to Watson and Crick. These guys built a full-size model. You can see it here. They didn't really know exactly how it fit together. You can see here it kind of curves. It's not exactly how it would fit. It's more, the model is more vertical. But they are the first one to coin the term double helix. And they really needed the help of Chargaff, understanding the base pairs, and that A pairs with T and C pairs with G. And they somehow acquired the information that Rosalind Franklin was working on, the picture specifically. And once they knew information about the picture, they went back to their lab, built the model, and eventually were awarded the Nobel Prize. And so here you see Watson and Crick and Wilkins, who was working with Franklin at the time. Unfortunately, she died due to breast cancer before that Nobel Prize was awarded, and the Nobel Prize is not given out after death. So she really does not get credit for any of the work she has done up until late. And that is it on the discovery of DNA. So the key people are Frederick Griffith, worked with mice, identified things were transformed or passed on between bacterial cells. Oswald Avery, DNA is the stuff. Erwin Chargaff, A pairs with T, C pairs with G. Rosalind Franklin, X-ray of DNA. And Watson and Crick, finally, the double helix. And that is it on the discovery.